Hello everyone, welcome to our discussion on population genetics and evolution. In this particular session, we'll be discussing the different phenomena that you observe in a population like genetic drift, then founder effect, bottlenecking, then Hardy-Weinberg equilibrium, etc. And also the factors that affect Hardy-Weinberg equilibrium or gene frequency. So, to begin our discussion on population genetics, we'll start the discussion by defining a species and a population. You consider a group of organisms as they are belonging to a species if they satisfy three criteria. And the three criteria are: one is their morphological appearance, that is, you call a particular group as a belonging to a particular species if they have these three criteria in common. One is their morphology. Coming to their morphology, if you call a particular group or two individuals as belonging to a species, they should be morphologically similar. They should have similar morphology. So the appearance, they should have many similar, or they should be morphologically, their physical appearances should be similar. The second criteria is regarding their genetic constitution. They should have a distinct genetic constitution. Distinct in the sense that this group or the group which you consider as a species should have a distinct genetic constitution when you compare to another group. For example, if you take human beings, Homo sapiens, they are having a distinct genetic constitution in the sense that we are having the chromosome number 42. We are distinct from our relatives. We are distinct from another group. We are distinct from Drosophila, which is an insect. So, you call a particular group as a species when they're having a distinct genetical or genetic constitution. So these are the two factors that you consider in recognizing a group to be a species. One is morphological similarity. Second one is genetically distinct, and the third is the most important one, that is reproductively isolated. That is, this group should be reproductively isolated in the sense that they can breed and produce fertile offsprings. There are cases in nature where related species they interbreed, but the problem is that when when you consider a particular group as a species and when they reproduce, they will be producing fertile young ones. But in the other case, so there are many examples where in nature where two related species they interbreed and they give rise to viable young ones. The young ones are not the young ones are only viable. I didn't use the term fertile young ones. Fertile young one means those young ones are capable of giving rise to their next generation or next set of or the next round of individuals because they are fertile. But when you consider the other case, that means two uh, related species, they will be interbreeding, but they will not produce fertile young ones. The example is uh, the uh, the young one which is generated by or by the mating of a horse and a donkey. You get something which is called a mule. Mule is a viable individual, so it is having life, it is viable. The embryo develops, it give rise to an individual, the individual also live. But the problem with the individual is, or the problem with the mule is, it is sterile. So when you consider a group as a species, it should be morphologically similar, genetically distinct, and reproductively isolated. So, if a group satisfies these three criteria, you call that them as belonging to a species. And this particular group of when you have many of them, you consider them as a population. That is called as a population. So, a population is a group of individuals belonging to the same species and they can interbreed. they are having the property, or they're having the capacity to interbreed, or such a group is also called as such a group is usually referred to as uh, uh, a population. And coming to the terminology, which is a gene pool, the sum total of all the genes in a population. So we defined what is a population, and the sum total of all genes in a population is referred to as a gene pool of that particular population. So if you take the example which is given here, you can see that this is a population of frogs, and you can see that they're having some uh, difference in a color pattern. That is, this is a particular population, and there is variation in color. You can see purple colors and also red colors. So all this together, the genes of all this together is considered to be as a gene pool. As you can see here, you have the genes for green color, you have the genes for purple, and you have the genes for the red color. So together, so here we have represented only we have taken this as an example and represented only their morphological appearance, which is the color. But when you consider a gene pool, it consists of all the genes, every genes in that individual, or what are the variations. There may be variation in the genes. If you take the skin color, as we have discussed, there are many variations. If you take height, there is variation. So in every aspect, if you take hair color, so in every aspect, or in every gene in a particular population together, is considered to be the gene pool. That is a sum total of all genes in a population is called as the gene pool of that particular population. And coming to the next term, which is gene frequency, or allelic frequency. So, gene frequency or allelic frequency is a term applied to the relative frequency of an allele at a particular genetic locus in a population. So, it is a frequency, that is, if you are considering a particular gene, we know that the gene will have different alleles. Okay, and if you are considering the gene frequency or an allelic frequency, it is a term which is applied to the frequency of an allele at a gene locus in a population, and it is determined by counting how many times the allele appear in a population and then dividing it with the total number of copies of that particular gene. So that's why I said here you can see that if you take for an example, if you consider the frequency of the allele, we are going to find out the frequency or gene frequency or allele frequency of this particular A, uh, the gene which is called as the dominant A. So that particular the gene which is, for example, this A is a gene which is indicating flower color. Okay, and that particular A gene, as we all know, it will have different alleles. I'm taking only two alleles here, a capital A and a small a, which these represents the gene for flower color. Take it as an example. And now I need to find out the gene frequency of this particular allele, or the allelic frequency of this capital A. So what I do is I should have to find out, to determine the frequency of this one. I need to have the number of copy of allele A in a population, which will be divided by the copies of genes in the population. Okay, we'll consider an example so that this might be more clear in the next slide, which where you have an example here. So suppose you are having 100 individuals in a population. So here you have 100 individuals in a population. Out of this, 40 are homozygous dominant, which is capital A, and 40 are heterozygous, capital A, small a, and 20 are homozygous recessive, small a. So now we are going to find out the frequency of the gene capital A. Okay, so here we have the formula for finding out the frequency of allele A, which is the number of copies of allele A in a population. So the number of copies, if you consider the capital A allele, the number of capital A allele in this population, you have to consider two categories. This particular 40 is having, that is homozygous dominant, they are having two alleles. Each individual will have two capital A's, and here you are having a heterozygous, 40 individuals are heterozygous here. They also having this capital A in them. So if you take the number of copies of capital A in this population, you have to consider both homozygous dominant as well as heterozygous. So in homozygous dominant, because each individual is having two copies. So here we are going to count the number of copies. So homozygous dominant, each individual will have two copies of capital A because they are homozygous dominant, and hence this will be, the number will be 40 into 2 because each individual is having two copies of capital A, so that is 80. And here in heterozygous, you have 40 individuals, but only half is capital A, the other half is small a. As a result, the number of capital A in 40 individuals will be 40 itself. So this particular, if you substitute here 100 individuals, each individual is having two copy because it is a diploid individual. Each individual will have two copies of that particular gene. As a result, the total number or total copies will be 100 into 2, which is 200, because each individual is housing uh, two copies of that particular gene, either capital A small a, or small a small a. So if I take a particular individual, if I take an individual, for say this is the individual I consider in a population, in this particular population, okay, and this particular individual in this particular population, he may have two copies of this gene. Either he will have two capital A's like this, or he may have two small a like this, or he may have capital A small a. So this is the possibility. And the number of copies of a gene in a population, we have 100 such individuals, and each individual is having two copies of this allele. Okay. As a result, if you take the total number of copies of the gene or the allele, it will be 200, 100 into 2. That is why you get 200. And this is how we explain this. That is, you have 120, which is divided by 200, will give you a ratio or the frequency which is zero point 0.6. The same is applied for if you have to find out the frequency of the gene which is called as small a. We have 20 individuals which are homozygous recessive. So 20 into 2, as we did for homozygous dominant, because each individual will have two copy. So that is how you got 40 here, 20 into 2. And the other, it is the same, because heterozygous individual, each individual will have one copy of small a, so that is 40. And here you have 80 by 200, which is zero point 0.4. So the allelic frequency of capital A is zero point 0.6, and the allelic frequency of small a in this particular population is zero point 0.4. So this is what is called as gene frequency or allelic frequency. So here, as we said, it is a term which is applied to the relative frequency of an allele at a gene locus in a population. Okay, so we have completed, we have discussed these two terminologies, what is a gene pool and what is a gene frequency. And now coming to the other terminology, which is or another phenomenon which is seen in a population, which is called as a gene flow. So gene flow is, it is nothing but it is migration. So when you talk about individual organisms, we call or we term it as when one organism or a group of organisms migrate from one place to another, or if they move from one place to another, you call it as migration. If they are coming into some place, you call it as immigration, etc. So the gene flow is the migration of genes. When individuals move from one place to other, the genes are also moving with them from one place to other. Not to confuse with migration. Migration is the movement of individuals, and along with the individuals, genes are also moving, and the movement of genes is called as gene flow. So when gene flow happens, what happens is that if you take, for example, we'll take this particular example which is shown in this diagram. So you have two populations here, two populations of beetles. So they are belong to the same species, but the thing is that they are having different color pattern or color. One is brown colored and the other one is green colored. So they belong to the same species. One for say one is they are being isolated from one place to other because of a mountain or because of a very big forest between them, etc. Whatever the case may be, but you can see that some of the individuals they start to migrate from this particular population to this. So here you can see individuals are migrating to the other population, and they come to this population, they interbreed among the individuals. As a result, this brown gene will be populated among the green. The same thing can happen vice versa. The green ones can move to this particular place and they can interbreed here. As a result, this green color or the green gene will be populated among this particular group. Okay, and if this has happened continuously for many generations, after a long period of time, these two population will have the same gene frequency. If you take the gene frequency before migration or before gene flow, so here this individual as take this particular individual as absent. This brown is absent here. So here, if you take the gene frequency of green gene or green allele will be 100 percentage or one, and here the gene frequency of this particular brown in this particular population will be 100 percentage or one, because only brown is present here. But if gene flow or migration occurs, and as I said, after migration, migration happens in is happening in both direction, and due to this particular migration, which is happening in both directions, uh, after after many years, after a long period of time, what happens is the gene frequencies of these two particular population will be same. That means because this brown gene is being populated here, the same green gene being populated here. Now, at the present time, you can say a clear distinction between this population. You can see that if there is a mountain between them, you can say that the beetles which is present in the left side of the mountain is green, and the beetles which is present in the right side of the mountain. So, if consider this arrow as a mountain, right side of the arrow is brown. After many years, if gene flow is happening, if migration is happening, after many years, you come to the same place. You are observing the beetles which is present in the right side of the mountain and also in the left side of the mountain. You can see, you will find that the number of green and brown in the right side will be almost similar to the number of green and brown in the left side, or left side and right side will have same number of or same gene frequencies. If you take the gene frequencies of both the sides, you can find that they will have almost the same gene frequency, and this happens because of this particular migration, and due to migration there will be gene flow. So the introduction of new alleles through gene flow increases the variability within the population and makes possible for new traits or combination of traits. So this is what happens here. So if the gene flow is high enough, the two population will have equivalent allele frequency and therefore can be considered as a single effective population. So At the present time, you consider this as two different population because they are having color pattern or color, their morphological color is slightly different, even though they can interpret. But when you come, or when you are having a very high level of gene flow, very high level of migration happening between the individuals after a pretty long period, you can consider them as a single effective population, because there will be the same number of green and brown will be present in the right side as well as the left side, and this is what is called as. So in simple terms, if you define gene flow, it is the migration of genes which causes introduction of a new genetic material in a population, or genetic exchange happens between two different population. And because of this, there will be change in the gene pool. Because if you take this as two gene pools, this was a gene pool which were having a different type of gene frequency. This was having a different type of gene frequency, and migration or gene flow will cause a change in this gene pool combination. Okay, so with this, we come to the end of the discussion of gene flow. Next is the other important concept, which is genetic drift, or the Sewall Wright effect, and the two variations of genetic drift, which is founder effect and bottleneck effect. We'll discuss both these in the next session. Thanks for hearing.